So now you know that the three events in sexual reproduction are pre-fertilization, fertilization and then post-fertilization. We have already covered the topics present in pre-fertilization, and just now we covered the topic double fertilization. The third the third event is post-fertilization. Post-fertilization implies what all changes happens to the parts within the flower after fertilization. Of course, from the previous chapter you know that after fertilization the petals will dry and fall off, stamens also will dry and fall off. What about the other parts? We will start with the egg. The egg after fertilization will get transformed to form zygote. So what will happen to zygote? This we will be learning in post fertilization events. Then we also learned that as a result of triple fusion, the central cell joins with the second male gamete to form a primary endosperm cell. So that means a central cell after post fertilization will become primary endosperm cell. Next, what about the ovule present? The ovule which is present around the embryo sac, what will happen to the ovule? The ovule will be transformed to be called as seed, which will later generate, and the embryo will grow up into a full plant. What happens to the ovary after fertilization? The ovary will turn to become fruit. Then we will be learning about the different types of fruits. So we will learn each of these in detail in post fertilization events. We will start with the zygote. The zygote has to divide and they have to develop into embryo. This process of division of zygote and differentiation into embryo is called embryogeny. Now there are two types of seeds. There is a monocot seed, that is monocotyledonous seed, and there are dicotyledonous seed. Monocot seeds examples, we can see monocot plants like banana, they are monocot, maize, rice, wheat, grasses, they are all monocots. We can distinguish monocots from dicots by looking at their leaves. They will have parallel venation, and also there will be only one cotyledon inside the seed. There are other differences too. In case of dicots, the leaves will have reticulate venation and they will have two cotyledons. Most of the plants are dicot, like beans, peanuts, coconut, all of them are dicotyledonous plants. Now the embryogeny, that means the transformation of zygote to embryo. Embryogeny in the initial stages is similar in case of monocot as well as dicot plants. So we will begin with the embryogeny. What you have to study in textbook in detail is the embryo formation in dicot embryo. See how the embryo forms from zygote. In case of a dicot, there you can see two cotyledons. So embryo develops from a diploid zygote, and zygote is unicellular. The zygote will divide to form a basal cell and another cell which will later form embryo. So this structure is called pro-embryo stage. This is the pro-embryo stage in the embryonal development of a dicot embryo. After the pro-embryo stage, the next stage is globular embryo. The basal cell will cut off a single layer of linearly arranged cells. These cells are called suspensor cells. This is the basal cell. And then the embryo will also divide to form a group of cells, and it looks globular, and so the name comes globular embryo. So what is the role of the suspensor cell? Suppose if this is the embryo sac, this is the embryo sac. You know that there are endosperms formed as a result of fertilization. So here you will find endosperm, primary endosperm cells. And you know endosperm is filled with food reserves. So the suspensor cell's function is to push the embryo towards the surrounding endosperm tissue, so that the globular embryo or the embryo can derive nourishment from it. So that is the role of suspensor cells. But as the embryo starts developing, the suspensor cells size will also reduce and it will get dry. The next stage after the globular embryo is heart-shaped embryo. The globular part of embryo will become heart-shaped. So these heart-shaped, these two parts are actually developing to form cotyledons. This is a dicot, so there will be two cotyledons. Cotyledons also serve the purpose to nourish the developing embryo. And then you can also see the size of the suspensor cells gets reduced by the time. So this is the picture of a heart-shaped embryo stage. As the embryo derives more and more nourishment from the endosperm, the size of the embryo grows, and the final stage is called the mature embryo. In the mature embryo, we can see the cotyledons are little more grown up. So these are the two cotyledons. In between the two cotyledon, we can see a part, a tissue. That tissue is called epicotyle tissue. So epicotyle tissue later will form leaves, and epicotyle tissue will terminate to form the plumule. Plumule is a shoot of the plant. So they will grow up to form leaves and all the aerial parts of the plant. Whereas the bottom part of the embryo will have a tissue called hypocotyle. Hypocotyle tissue actually forms a stem of the embryo. So if you imagine this as the future stem, and if you imagine this as the embryo axis, the hypocotyle will divide and form the stem. The epicotyle will develop to form the leaves, and the plumule will become the shoot. So behind the hypocotyle will terminate below as the radicle, and the radicle develops to form the root of the plant. And the suspensor cell will gradually diminish and they will vanish. So this stage of the embryo is called mature embryo stage. Mature embryo. So these are the stages in embryo development of a dicot plant. In embryo development, the initial stages, that zygote as well as a pro-embryo as well as globular embryo, they are common in monocot as well as dicot. But after that the changes comes. In case of dicot, there will be two cotyledons. The first stage in the embryogeny is zygote will divide to form a pro-embryo stage, where there is a basal cell and the future embryo cell. The basal cell cuts off more cells serially and they are called suspensor cell, and the embryo divides further to form a globular shape. Suspensor cell pushes the globular embryo towards the endosperm so that they can derive nourishment. Endosperm is filled with food reserves. Globular embryo will divide further and become heart shaped. So, this stage is called heart shaped embryo. Actually, the heart shape what you see, they are cotyledons. The final stage is called mature embryo, where the tissue which is present between the two cotyledons is called epicotyle, which will later develop new leaves and they will terminate in plumule, the shoot of the plant. Below the level of the epicotyle, you can see the hypocotyle. This tissue will become the stem of the plant, and hypocotyle terminates at the end by radicle. Radicle develops into the root of the plant. So, that is how the embryo develops in case of dicots.